La diffusion vient de démarrer. Tous les participants sont en mode écoute seul.
Good morning, everyone, and welcome to this webinar. Thank you for attending today. We will be discussing our next generation sequencing library preparation solutions for challenging samples. If you have any questions during the webinar, please type it in the chat box, and then we will be taking them at the end of the webinar. So let's get started. I hope you can hear me well. All right. So today we will be discussing three types of challenging samples. So first I will be talking about how our smart technology has allowed to do single cell RNA sequencing and what are the benefits of using this technology. The second time I will explain how we can also work with samples that are both of low quality and low quantity using the smarter stranded PICO-V2 technology. And I will show some examples using FFPE and circulating free RNA samples. And we will finish by discussing also our solutions for DNA-seq using also challenging samples like FFPE, chip-DNA, and circulating free DNA samples. So let's start with single-cell RNA-seq, and before that, just to briefly explain why is this field getting more and more importance and why people are more interested in single-cell sequencing. So there are several studies now that have shown that actually the gene expression is very different between different cells. And when you are taking a bulk of cells, you will be analyzing only an average of the expression. So this is what we can see here in this figure taken from the papers cited below. And here the authors, they compare what they obtain using 10,000 cells here in green compared to using single cells that you can see here in blue. So for some genes, the patterns of splicing and expression is similar between the bulk population and the single cells. But for some others, you can see that there are very big differences between different cells. And these cell-to-cell differences have been shown to be critical in many physiological processes and also in diseases. But doing single-cell RNA sequencing is not easy, and there are many challenges associated with this. And this comes from the fact that actually the amount of RNA that is available per cell is quite low. So it's estimated that one mammalian single cell has in average 10 picogram of total RNA. And from this, only 1% approximately corresponds to messenger RNA. So you can imagine that the amount that is available is extremely low. So for this, you will need a method that is sensitive enough to perform the cDNA synthesis from this small amount of RNA. The method must also be reproducible because you want to see the biological variation and kept to the minimum the technical variation. And ideally, you should be able to use the cells directly as input. And this is to avoid any RNA purification. And of course, you will also want to have minimal handling to limit the sample loss and degradation, contamination issues. So all these features is what we propose with our smarter technology, as we will see. So here you have in this cartoon the workflow for our SmartSeq V4 ultra-low input RNA kit for sequencing. This is what we use for single-cell RNA-seq. This kit can work directly from cells, so you can use 1 to 1000 cells directly as input, or you can also start from purified total RNA. Since we are using oligo-DT priming, no removal of ribosomal RNA or no DNase treatment is required. You will only be amplifying messenger RNA. All the workflow happens in a single tube, and at the end you will get full-length cDNA that you can process for ion torrent or Illumina sequencing. So let me explain a little how this technology works. So you will start with from your total RNA or your cells. The reverse transcription will be started by this oligo-DT primer, which contains a sequence. This is called here in green the SMART sequence. And we use a reverse transcriptase that when it reaches here, the 5' end of the mRNA, it will add some non-templated nucleotides represented here with X's. We have characterized these nucleotides and we perform this reaction in the presence of another oligo, the SMART-SICV4 oligo, which contains nucleotides that will anneal to this non-templated one. This allows the reverse transcriptase to switch the template, so to use this oligo as template for reverse transcription, and it will copy this other sequence, SMART sequence. So at the end, what we will have is a double, we will have a first strand cDNA with known sequences at each end, and we can then use a primer to amplify this, and at the end having double-stranded cDNA with known sequences at each end. Well, I'm sorry here, the double-stranded cDNA for some reason is not showing, but this is what is obtained, and here we can see a bioanalyzer trace showing what you typically obtain from human samples from different inputs, you can see here, and you will obtain cDNAs from around 400 base pairs and up to 14 kb. So you will really capture the full length cDNA that you have in your samples. So this full-length cDNA, as I said, needs to be processed to be used for sequencing. So for this, you will need to fragment into pieces of the appropriate size for sequencing, and then add the adapters depending on the sequencing platform that you will use. So these are the recommendations that we provide in our user manual. For ion torrent sequencing, we recommend doing an enzymatic sharing and then use Thermo Fisher Ion Express Library Prep. For Illumina sequencing, there are two options. Either you can use a mechanical fragmentation using Covaris, and then use our triplex DNA sequencing kit to add the Illumina adapters. Or you can use the Nextera XT product from Illumina, which uses segmentation. So this product will simultaneously fragment the cDNA and incorporate the Illumina adapters. So you have these two options. As you notice in the name of the kit, this is the fourth version of this technology that we have been improved year over year. So here, just to show you a comparison with the previous version, the Ultralow V3, using 10 picogram of mouse brain RNA, which is the smallest input amount that we recommend. We did the sequencing in duplicate, as you can see here, and we notice that we are more sensitive because we are able to reduce the number of cycles, and even in doing that, we are identifying between 30 or 40% more transcripts, as you can see here, as compared to the previous version. So we have improved the sensitivity, but also the reproducibility. This can be also seen here when we compare these plots in which each point represents a transcript in one of the different replicates. So the closer these points are, the better the reproducibility is. So you can see that in general, if you take the overall transcripts, those that are here in the upper parts, which are the most abundantly expressed, they have a quite good correlation. But when you go here to the lower part, so the lower expressed transcripts, you can see if we zoom here, that the correlation is not that good. So this was the case with the previous ultra-low V3, and this we were able to improve with the version 4. So there were many modifications, improvements to the V4, but the most important of this was incorporating LNA technology, log nucleic acid, in our template switching oligo, and this, as we could see, had the effect of improve the sensitivity and reproducibility. This SmartSeq technology for single cell is available in two formats, let's say, or for two different applications. You can use the classical SmartSeq V4 kit to generate full-length cDNA. As is shown here, you will have a transcript with coverage from 3' to 5'. Here, for example, we are comparing different single cells. You can see the coverage is very even. You will use full-length cDNA when you are interested in doing annotation of complete transcripts or interested in using splice variants. And of course, you can use also the data for differential expression analysis. And this technology is available in many reaction sizes and is also available for the Fluidigm C1 system, which is an automated platform for doing this single cell cDNA preparation. We also have SmartSeq technology only for the 3' end of the transcript. So this is the SmartSeq V4 3'DE kit that will allow you to capture only the 3' end of the kit. So this is when you don't need the full transcript. You are not really interested in splice variants. You only need to compare the expression in different cells. So this you can allow this kit. This also will reduce cost, and you will be able to sequence more samples per lane for reducing the cost. And here it's important to note that the input is a bit lower, so you can also start with one single cell, but up to 1000 cells can be used, and from 10 picogram to 1 nanogram as compared, to 1 to 1000 cells for the original before, and 10 picogram to 10 nanogram of input RNA. So this technology for single cell sequencing has been used extensively and with many different cell types. As you can see here, just some screenshots of the papers in which this technology has been used. The use of cells directly as input has only been validated with mammalian samples, with mammalian cells, because this kit uses a lysis buffer that is able to lyse these mammalian cells. However, we have not tested if this kit is strong enough to lyse some cells more difficult, like is the case of plant cells or yeast cells that they have a cell wall. So in those cases, we prefer to recommend that people extract the RNA and then use the RNA as input for the kit. So what we know for sure that will not work with the SmartSeq V4 is two types of samples. Is low quality RNA samples and non-polyadenylated RNA samples. And this is because the SmartSeq V4 utilizes oligo-DT priming. So it means that the there must be polyadenylated RNAs to allow the reverse transcription. And well, this is not the case in bacteria samples. They are usually non-poly A first. And also in just eukaryotic samples that normally should have a poly A, but when they are degraded, degradation of the RNA usually starts by the extremities. So what happens is that usually this degraded RNA does not have anymore the poly A tail, so you cannot use this technology. So this leads us to the second part of the presentation, what to do if you have this kind of degraded samples, as you can obtain, for example, for FFPE or from circulating free RNA. So when you have these low input and low quality samples, you will need to use random priming instead of oligo-additive priming for the reasons I previously explained. The problem here when we use random priming is that we will also be amplifying ribosomal RNA. And ribosomal RNA accounts for around 90 to 95% of the total RNA. And you don't want to sequence this, you want to get rid of that. There are some products in the market, and we have one for that, for depleting the ribosomal RNA from total RNA samples. This is the Ribogon RNA, Ribogon mammalian, sorry. But this requires at least 10 nanogram of total RNA, as most of products for this type of depletion. So we wanted to also offer a solution for people which didn't have as much as 10 nanogram of total RNA. And this is what I am presenting here, the Smarter Stranded Total RNA-seq kit Pico Input Mammalian V2, but I will refer to it only as the Pico V2, it's shorter. So this is a very nice solution that will allow to do total RNA-seq from as low as 250 picogram of total RNA. So as I say, in up to 10 nanogram of total RNA. As I say, when you have more of 10 nanogram RNA, there are other solutions that you can use. But this is a bit complicated when you have less than 10. So this product will allow you to do that. In the Pico V2 kit, we use a different technology, a novel proprietary technology that allows to remove not the ribosomal RNA, but the ribosomal cDNA. So what we can see here in this workflow is that we are going to do the reverse transcription using random primers, as shown here in N6 primers. And we are going to amplify all the RNA in the sample, so including the ribosomal RNA shown here in purple. Once that we have this cDNA, we will do an incubation with an enzyme called ZAPR and some probes called rProbes that will target specifically the cDNA which was reverse transcribed from ribosomal RNA. And then we will have PCR step in which we will enrich the sequences that were not cleaved at that step, so those that are not ribosomal cDNA. So this process allows us to, at the end, have a library which is enriched in the sequences which are not ribosomal RNA. Of course, we will see that some of these ribosomal cDNA sequences can escape to this degradation, but still we're able to get rid of most of it. Another important feature of the Pico kit is that it's compatible with RNA of any quality. So if you are familiar with the RIN scale, which measures RNA quality and usually goes from 1 to 10, and 10 is a high quality and 1 is low quality, you can see so that the Pico V2 is compatible with almost any quality of RNA. So even with RIN as low as 2. During the workflow, we have the incorporation of the Illumina index and adapters. It means that you don't need to buy a separate library prep kit. You will start just with the RNA, and then you will end with your library, which is ready to sequence. And the whole workflow is quite streamlined and fast. In only 6 hours, you are ready to sequence. The protocol also provides strand information. So if you're interested also in studying non-coding RNA, this is something that is useful. And in the Pico V2, as the name indicates, is an improved version of the version 1 that we had before. And we had made some improvements that are mainly related to increase the compatibility with all Illumina platforms. So this new version has full compatibility even with the latest Illumina sequencer, which was not the case with the version 1. Had some problems to work with the HiSeq 3000 or 4000. This is not the case anymore with the V2. It's fully compatible. So let's see some data generated with this kit. We took here mouse total RNA from brain, and we tested it in duplicate with different amounts of RNA. And you can see here that we tested even beyond the recommendations of the kit, so with 50 nanogram and as low as 100 nanogram. And what's interesting to see is that no matter the input amount, the number of transcripts identified is quite similar. The reproducibility is also very good, as we can see here, because as I said, they were done in duplicate. And the strandness is very good. And the depletion of ribosomal RNA is efficiency between 85 and 90%, depends. So you can see here the amount of ribosomal RNA that's left. We can also compare here. This is also quite impressive that if we compare across the input amount, so a very low input amount as 250 picogram and a high input amount as 50 nanogram input, the reproducibility and the correlation between these two samples is very good. So this shows the robustness of this approach. But so this experiment that I showed before was done with good quality RNA, which was dilute to different amounts. But we were discussing that sometimes you don't have this nice RNA. You may be interested in working with more difficult samples like FFP. So we did this experiment in which we use 4 different FFP tissues. And you can see that the quality is very variable. So what we are measuring here is the DV200 value, which will let you know which is the percentage of your RNA sample, which are of higher size than 200 nucleotides. So you can see that we tested with as low as DV200 of 28%. And you can see that in all the different cases, we were able to prepare a nice library with the Pico V2. In terms of sequencing, here is what we obtain. So we can see that for each sample, actually, we took a small amount, 10 nanogram, or a high amount, 40, 72, 50, it depends on the sample. And all the experiments were done in duplicate, and it's very interesting to see that the number of transcripts identified is quite reproducible between the duplicates and also between the different input amounts. So this shows that you don't need to put high quantities of your FFP sample to get good results. With as low as 10 nanogram, you already are able to detect most of your transcripts. We also show data on our website in the tech note with lower than 10 nanogram. It can be possible with the FFP sample to use as low as 1 nanogram, but for best results we recommend to use 5 of 10 or 10 nanogram if possible. The correlation is also very good when comparing the low input and the high input, and this even using a very degraded sample like this one, sample number C, which remember has a very low DV200. Another application for this kit is the use with cell-free RNA-seq. Sorry, with cell-free RNA samples. So cell-free RNA is also usually low abundant and quite degraded. So this is actually data done by a collaborator. And this team, they wanted to push the limits of the kit, and they compared Pico version 1, so the previous version of the kit, with the current Pico V2 version at two different input amounts, so 1 nanogram and 150 picogram, which is below the specs of the kit. And they did these libraries in duplicate. So what they saw is that overall, the Pico V2 provides better sequencing information, and this is more important at the lowest input amount, so 150 picogram. So we are obtaining at this low input amount a higher number of a higher mapping rate, sorry. And also we are decreasing here the amount of reads mapping to ribosomal and mitochondrial sequencing. And the number of genes detected is increased here, especially in the 150 picogram. So this data show that with the Pico V2 you are increasing the detection, especially when you are working with very low input amounts. Here there are some only examples of publications obtained with the previous version. This Pico V1, the Pico V2 was just launched, so we don't have yet the publications. And just to show also that there are many type of samples that can be used with this kit. So many people have used FACS sorted cells to analyze specific cell populations. Also, they have used nuclei or laser captures microdissected samples, and of course, can be also used with FFPE and cell-free RNA, as I just mentioned. So the last part will concern now DNA. So we are moving from RNA samples to DNA samples, and for this we are using a different technology called Truplex. And we will see that it can be used with other also challenging samples like FFPE, chip DNA, and circulating free DNA. So what are the characteristics of these challenging DNA samples? So first, for FFPE DNA, usually the quality of this DNA that is isolated is expected to be low, and it will generate low complexity NGS libraries because the usable amount of DNA is low, despite an apparent concentration of high amount of DNA. Actually, the amount of DNA that you can use for library prep out of that is low. So similar to what we saw with RNA, is better to put higher amounts. But sometimes the amount that you recover is just too small. Another application in which you will recover low input, low amounts of DNA is chip chromatin immunoprecipitation, because you will only be recovering the regions that are bound by a protein or a complex. And if besides of that you are starting with a low number of cells, you will get even lower DNA amount. And the third type of challenging sample is cell-free DNA. So this is typically isolated from plasma or serum, but also some people are isolating this from urine, cerebral spinal fluid, or CSF. So this cfDNA is typically fragmented, of small size and low abundant, but this is of course very variable depending on the patient and the disease. But this is also then a low abundant sample. So what we offer for this challenging low abundant samples, triplex technology, which has the benefit of being also a single tube solution, very fast. It's only 3 steps with 15 minutes hands-on time. There is no any intermediate purification step between these 3 steps, so there is no risk of loss of sample in any of these steps. So you will just put your sample in one tube, and in 15 minutes hands-on time and 2 hours, you will get your library ready to sequence. So this is very briefly the workflow of the triplex kit. You will start with double-stranded cDNA. It must be fragmented. You don't need to do fragmentation if it's cell-free DNA, because as we said, it usually is of short size. And triplex, as I said, incorporates 3 steps. So the first one is the end repair, the fragmented DNA. The second is the ligation, and here we are using stem loop adapters that are ligated with a high efficiency, and also they have this feature that they contain a cleavage site, which allows after the extension to eliminate these adapters that were not used, and this will tremendously reduce the background. And also you don't need to do any titration, as is the case with other kits. You will need, with some other kits, you need to titrate the adapter amount depending in your own input amount. This is not necessary with triplex. There is no optimization step. And then there is a PCR step in which the Illumina indexes are incorporated. So you will get indexed libraries, and we provide indexes to do up to 96 dual indexed samples. Everything is included in the box. This technology is available in 3 different formats. So we have the original triplex DNA-seq, which is the kit that you can use for any DNA-seq need. So you can use it with FFP DNA and cheap DNA, importantly, and you can see that it's very sensitive. So it works with as low as 50 picogram of input DNA and up to 50 nanogram. This chemistry was optimized in the triplex plasma-seq kit for people working with circulating free DNA. You can see here that in this case you will need to use from 1 nanogram to 30 nanogram of cfDNA as isolated from a liquid biopsy. We have also people that have tried less than 1 nanogram, and they still can get good data. And the latest that we have developed is the triplex TAG-seq kit. So it's a similar chemistry, but unique molecular tags are added in the adapters, and so they are incorporated in the DNA fragments during ligation. And this is useful for people that are interested in detect rare alleles that can be as low as less than 1% of frequency. And in this case, you will need also, we recommend 1 nanogram to 50 nanogram of input. So just to show you the kind of data that you can obtain with FFP DNA. So here we use 9 samples of FFP DNA. So either from healthy people here in NAT, or from tumors. Writing here as malignant. We tested these 9 samples with our triplex kit and compared to competitor A and B. And we can see that triplex DNA-seq libraries provide the more diverse libraries in all cases. In terms of duplicate, we are keeping this very low with triplex, which is also something that you want to have in your libraries, low duplicates. And in terms of GC coverage, we can see here that triplex provide the more uniform GC coverage as compared to other kits. This technology, as I said, can be used also with ChIP-seq, and it has actually quite used in many different publications. So in our website, we have a specific section dedicated to that. You will be able to see all different models and applications in which triplex has been used with ChIP-seq. So in cancer studies, stem cell studies, developmental studies, etc. With cell-free DNA, also there are very nice publications that have used our kit. So in this one, for example, triplex was used to prepare NGS libraries from cell-free DNA, and the libraries were enriched with protocol for enrichment and only sequencing whole exome from circulating tumor DNA, and from cancer patients and also from healthy donors. And with this study demonstrated for the first time this the ability to monitor a tumor evolution before and after treatment, and this using only blood samples. So this is a field that is getting more and more interest because it provides the possibility to test and to monitor cancer progression or cancer markers using only blood without need to take tumor biopsies directly. Another interesting application is for this cell-free DNA using triplex plasma-seq is in non-invasive prenatal diagnostics. So, for example, in this paper, it was the first study that demonstrated that it could be possible to determine the genome of a fetus only using the circulating fetal free DNA found in the pregnant mother. So in this case, triplex was used to prepare a library using the DNA extracted from the maternal plasma. So to conclude, the benefits of the triplex approach are that you are using a single tube. This will allow you to preserve the sample material and then the library complexity. You will be minimizing any handling error, sample mix-up. The protocol will be much faster and convenient. There's no optimization required. It works the first time and every time with high reproducibility. And of course, importantly, it's very sensitive. So this is important when you are working with limited samples. We have seen that it can work depending on the application from 50 picogram of DNA or from 1 nanogram. And there are many publications and studies that have proven the utility and performance of this product. So if you want to learn more about any of the different kits and solutions that were presented today, I invite you to go to our NGS resource portal at www.takarabio.com/NGS. We have there many more information and for these kits and also for other solutions that we have. So with that, thank you very much for listening. And now I will check for the questions. Okay. Let me check that. So Yes, so as I say, you can continue sending your question in the chat box. So there's a question here. If what happens for the picoRNA, I say that it could be used with the RIN to 2 to 10. So if there is a difference in the protocol, so the answer is yes, you won't treat in the same way if you are starting with a high quality RNA than with a degraded RNA. Actually, the protocol provides two options, and there is one option with fragmentation and another option without fragmentation. So if you have good quality RNA, you will choose the option with fragmentation, and actually this is only an incubation step at 94 degrees, 2, 3, or 4 minutes. It depends of your sample, the quality of your sample. And that's it. So this is the fragmentation for good quality samples. If you have FFP samples or degraded samples, you will go for the option number 2. This is the protocol without fragmentation. So yes, this is a good question. It's important to follow the appropriate protocol depending on the quality of your input amount. Another question here again regarding the Pico P2 is that if it can be used directly from cells. So, well, all the data that I show here concern the use of the Pico V2 using purified RNA. So this is, let's say, then the normal specification of the kit is start with RNA from mammalian and at least 250 picogram. However, we have done tests in-house using cells directly as input for this kit. So similar to the SmartSeq V4 that I showed for single cell. And actually, we are using the same lysis buffer from SmartSeq V4. We have used it to process cells and put them directly as input for the Pico V2. So if you are interested in this approach, we have an alternative protocol that we can send to you. But this alternative protocol is not yet for single cell sensitivity. We have seen that it works with 50 cells, it can work. So yes, if you have 50 cells or more and want to skip RNA extraction, you may be interested in trying this protocol. Okay, another question here is if the Pico is available for other than mammalian. So no, at the moment this technology only works with mammalian samples. The reason is that the probes that are used to target ribosomal RNA, they only target mammalian RNA. We have tested it with mouse, human, and rat, and we think that with other mammalian there should be no problem, but we have not tested it with other species. We had feedback from a customer who used that with zebrafish, and at least in his hands, he was satisfied with the results, but it's difficult to recommend beyond the specifications of the kit. So at the moment, we just recommend for mammalian. Okay, I think we can stop here. I don't see other questions. Okay, so if you have other questions after this webinar, please do not hesitate to write us to tech support. So the address is techeu@takarabio.com, and we will be happy to answer this for you. Thank you very much, everyone, for listening, and have a nice day. Bye.